cycle.
Good evening, ma'am.
Can you, good evening, can you hear me?
Yes, ma'am. I can hear you.
Okay.
You can share your slide.
Yeah, just I'm adjusting the microphone. Yeah.
Okay.
Acha. First, I should share. Where is the share? Present now.
Present now.
Your entire screen. Are you seeing my screen? No, I have to click here. Yeah, share. Now.
Yes.
Okay.
Yes, ma'am. It's visible.
Okay.
So, ma'am, we'll start at 7 exactly.
So we'll let this be on?
Yes, ma'am. Can.
Yeah.
Good evening, everyone. Welcome to the Progress and Prospects in Biology special lecture series. Thank you again for joining us from all over the globe. Now, before we start, you must be aware of this by now. There are a few housekeeping rules that I would like to remind you. Please make sure to keep your microphones muted, your videos off during the lecture to preserve the bandwidth, and do not share your screens at any time. Otherwise, we shall be forced to remove you from the meeting and not readmit you. I request everyone to keep the presentation window pinned to your screens so that we can continue uninterrupted. Please post your questions in the chat box in both Google Meet and YouTube, and we shall address them at the end of the lecture. If you have any questions that are not related to today's talk, please feel free to email us. And the link for the feedback form shall be given towards the end of the meet, and it will be open till 8:30 p.m. only, and it will not be shared by email. So anyway, today we are honored to have with us Professor Chandrima Shaha, Professor of Eminence at the National Institute of Immunology, New Delhi, and the present President of the International Science Academy. So now I would like to request our mentor and convener of the series, Professor Renare Banerjee of the Department of Zoology, University of Calcutta, to welcome and introduce our speaker. Over to you, ma'am.
Thank you. Can you hear me?
Yes, ma'am.
Okay. Chandrima, what can I say? It is a proud moment for me to be welcoming you on this webinar series. And not only because you are one of the sharpest minds in science of country today, but also because we are privileged to share the same school as our foundation and beginning. So today I welcome everyone, all the participants on the Google Meet, and there is a live streaming on Facebook too, as well as the YouTube live streaming for Professor Shaha's lecture. I welcome all the participants. For those who are our regular viewers and participants, you know that Progress and Prospects in Biology webinar series was conceived and is maintained and executed very smartly by a group of PhD students and postdoctoral fellows, just like yourselves. And we have undergraduate and postgraduate students, scholars, teachers, scientists, junior to senior professionals tuning in regularly to the various scientific talks. So today we have with us our speaker is Professor Chandrima Shaha. Her primary interest in research includes the understanding of the intricacies of programmed cell death processes in rapidly proliferating cells. She is the former director of National Institute of Immunology, New Delhi, where she is currently the Professor of Eminence. She is also the President of the Indian National Science Academy. She is an elected Fellow of the World Academy of Sciences and of all the 3 academies in India. She served as Vice President of International Affairs of the Indian National Science Academy and as a member in the councils of all 3 academies. Notable awards include the Ranbaxy Science Foundation Award, the J.C. Bose Fellowship, the Insar Shanti Swarup Bhatnagar Medal, D.P. Verma Memorial Award, Om Prakash Bhasin Award, Archana Sharma Memorial Award of the National Academy of Sciences, Darshan Ranganathan Memorial Award, and the Chandralekha Hora Memorial Medal of the Indian National Science Academy, and the Shakuntala Amir Chand Prize of the Indian Council of Medical Research. She did her MSc from Zoology Department of the Calcutta University and PhD from the Indian Institute of Chemical Biology with postdocs at the Kansas University Medical Center and the Population Council at New York. So that is a very, what should I say, illustrious career that Chandrima has left in her wake. And today we are privileged that we shall be able to hear another of her talks, and she has many researches to her credit. It is titled Alive and Well Within the Sentinels of the Immune System, the Protozoal Invader. So a very warm welcome, Chandrima, and we are all eagerly awaiting to hear your talk. And after your talk, we have some alumni from our school who would maybe like to interact with you. And this is also the centenary year of our department, Zoology Department, Baliban Science College, and also of our school, Gokhale Memorial Girls School. So this is a privilege for us to meet on this platform. So now, over to you, Professor Shaha.
Yes, it is indeed a coincidence that school and the zoology department, where I'm alumni of both. So it's a coincidence. But thanks to you, Anna. I mean, I would like to formally thank you for giving me this opportunity to come to this forum and talk perhaps to many young people who are there also. I'm not sure. I was not sure what kind of audience would I have, so I have sort of tried to make it as easy as possible of the work that we do. And since the expanse of the work is quite a bit, I have chosen some bits to present. And in these days, host-pathogen interaction is very important. And although I'm not going to speak about our nemesis, this coronavirus, but I'm going to speak about host-pathogen interaction. And in reality, what we have is that we thought actually that we had the control over the world, which obviously we don't have. We don't even have control over our bodies. And so it is, we are actually outnumbered. The microbes outweigh all visible plants and animals on Earth by a factor of 100 million. We have around us about 1,400 known species of human pathogens that include the viruses, the bacteria, the fungi, the protozoa, and the helminths. These are all known to the audience, I'm sure. But I would, ask for a little bit of indulgence for perhaps those who is not a biologist. Now, these bacteria are pathogenic bacteria, or even the non-pathogenic bacteria. They live in almost every place in the world, in deserts, geysers in very hot temperatures, poles in very cold temperatures, tropics, and under high pressure below the ocean, in the deeps of the ocean. So they are everywhere, and we are actually outnumbered in that sense. Now, there was an interesting question that how it will be in a world without microbes. Now, Louis Pasteur said that life would not long remain possible in the absence of microbes. So there were these famous experiments where people tried to make microbe-free animals and number of experiments. But this happened when Louis Pasteur said that at a time when it was realized that we have many useful bacteria. So if we look at the microbiome, if we look at this diagram, you will see that almost everywhere in our body we have bacteria. that are non-pathogenic, and some of them are actually useful. So our bodies have about 30 trillion cells. We are composed of 30 trillion cells. And but the microbes are 40 trillions in our body, which approximately weighs about maybe 3 kgs. And so we have a thriving ecosystem within our cells. And from the time we are born to the time we die, these bacteria are carried on in our body, and they keep on changing. The composition keeps on changing, and it has become such that it appears that every person has a unique microbiome signature. And even they're talking about now to lift these from the crime scene to match it with the microbiome of the criminal and identify him. So the microbiome is now a big thing, and these are all non-pathogenic mostly. But what do we do with the pathogenic bacteria? So of course we have to use our immunity. So there are two systems we have. One is the innate immunity, and the other is the adaptive immunity. Now, the innate immunity is the first attack on the pathogen. And it happens within about 12 hours of time. And the innate immune cells are like natural killer cells, the neutrophils, the mast cells, dendritic cells, eosinophil, basophil, monocyte, macrophage. Remember our blood reports, we find all these cells in the count. And of course, then the three main cells here who are able to phagocytose pathogens are the macrophages, the dendritic cells, and the neutrophils. The adaptive immunity is the second rung of immunity that spans from 1 to 3 days and even more if it is not successful, even more. So, but there is a connection has to be made between the innate immunity and the adaptive immunity. So these two systems have to collaborate at some point. So it is the dendritic cells and the macrophages who take in these pathogens, they digest them, and express their antigenic peptides on the surface of cells. And these cells are presented to the T cells of the adaptive immune system. And the T cells, the T helper cells, actually sense this and instigate the B cells to differentiate into plasma cells who generate antibodies, or T cells would activate CD8 T cells and the killer T cells who will kill the pathogens directly. So in very short, this is the kind of response that we give. And if you look at this schematic, you see that all the three, neutrophils, monocytes, or macrophages and dendritic cells, can kill pathogens by phagocytosing them and then taking it to fuse with the lysosome, which contains the digestive enzymes, and the phagolysosome is then degrades the pathogens and excretes them. Now, neutrophils are very smart cells, but they are very short-lived, and they are found mostly at the site of inflammation, and they can kill pathogens by phagocytosis. I'll come to neutrophils later. Monocytes and macrophages are phagocytic cells that can engulf and enzymatically break down microorganisms, as I said, and present it to T cells. So does the dendritic cells. So this may sound very simple, but it is a very, very complicated system that we have in our body. Now, how the pathogenic microbes are recognized? They have something called pathogen-associated molecular patterns, or PAMPs, on their surface. So these PAMPs are recognized by toll-like receptors on the cells and the macrophages and the dendritic cells. And then they are engulfed, and because they are actually recognized as non-self. At this point, I would like to go back a little bit to our microbiome and say that we have so much bacteria in the body. Why these pathogenic bacteria are being attacked by the immune cells, why they are not being attacked, why the useful bacteria are not being attacked. Now, you probably know that the T cells who distinguish between self and non-self are educated in the thymus. So once they come out of the thymus, they know in the body what is self and what is non-self. So after one round of education in the thymus, these cells are again educated in the gut to not to recognize the gut microbiome or other microbiota in our body as self. So there is this double education that happens. So this is how the pathogen recognition take place, and the other the non-pathogenic bacteria are not recognized. So same with the cells of our body, which are recognized as self. So now coming to pathogens, I am going to talk about a pathogen which is carried by an insect. Now, it is a very deadly combination, and there are many bugs that actually carry these bacteria, pathogenic bacteria, parasites, and viruses. And so, to name a few, you see here that malaria, dengue, chikungunya fever, Zika virus, yellow fever, West Nile fever, Japanese encephalitis, leishmaniasis, Chagas disease. So I'm going to speak about leishmaniasis, and you see, which is the leishmania parasite, which is carried by the sandfly, and it passes from the insect to the human. Again, from human to the insect, they have a growth cycle in the insects, and they go to again is transmitted to the human. There may or may not be intermediate carrier of an animal. So these are vector-borne diseases. So one of them I'm going to talk about today. So I just start with a little story that in the 1800s, early 1800s, 1824, 1820, around that time, this is a village of Bengal, and this is a drawing taken from the London News, and which was published at a later point of time. So a mysterious fever started occurring in these villages. That was in these 1800s, 1820s, when this was near about the district of Burdwan. So now, around 1862, fevers kept on coming, villagers kept on suffering, and particularly in 1862, what happened? There were villages after villages that got wiped off by this mysterious fever, and where the patients had a very emaciated look and had splenomegaly or hepatomegaly. Now, there were a lot of studies, and a study by William Twining, who was the assistant surgeon in General Hospital in Calcutta, and he wrote about the diseases of Bengal. He mentioned about this fever, although he didn't take any name. Then Dr. G.C. Roy, who was actually inspecting medical officer in Burdwan, named it as Burdwan fever. And the next year, almost the next year, Surgeon John Gay French, who was the first resident surgeon in the General Hospital at Calcutta, described it again as the Burdwan fever, where it was frequently confused with malaria. But malaria was different. So it didn't have the typical symptoms of malaria, but when the patients first came, they were treated. They were treated for malaria. So it was then called the Burdwan fever and the Kala Azar, or leishmaniasis as we know it today. So this disease actually created a lot of interest amongst doctors in the British setup, and two gentlemen who were in the British Army, William Leishman and Charles Donovan, they looked at smears. This is a smear from a person who died in Dum Dum, very near to Calcutta, and his histopathology was done of his spleen, and it was found that he had these little bodies that are marked here. And this was a rat which was infected with leishmania. They were showing these little bodies, which Leishman called them, trypanosomatid remnants. But Charles Donovan, who was another doctor in the Madras hospital, he said They are not trypanosomes, but they are something different than parasites that we have known. So in this raging controversy between the two, the British government asked Dr. Ronald Ross to actually look at this. So you have heard about Ronald Ross, if not about Leishman and Donovan. But Ronald Ross, what he did is he looked very closely at these, and he published a paper in BMJ in 1903, where he called this parasite Leishmania donovani, from taking the clue from Leishman and Donovan. And later on, when staining systems improved, the same bodies were looking like this, and when the Giemsa stain came in. So this was the story of how Leishmania donovani was named. And this disease kept on killing a lot of people until Professor UN Brahmachari came. And he made a preparation of antimony, which is stibinilic acid and its various salts. And he used that to treat people with Leishmania. And this is a graph which shows in Assam, where in the treatment of Kala-Azar, that this was very effective in taking down the disease. However, this was very toxic. But even today, antimony, right at the moment, miltefosine is being used, but very in the very near past, this antimony was used. Antimony, arsenic, all these heavy metals were used, but antimony was the most effective one. So we actually looked at the antimony in various situations, and just to say that there was a lot of resistance to antimony. So people started using mixture of drugs with amphotericin B and along with arsenic and antimony mixtures. So a lot of drugs were tried. What we found at one of our studies is that ergosterol is the most predominant sterol of the Leishmania parasite. And if there was low ergosterol, the parasites were very sensitive to this antimony. But if the ergosterol was more, they became resistant to antimony. So this could have been one of the reasons, and it has never been proven. But in the laboratory, we could prove that if you have more ergosterol, then the parasites become resistant. And all the resistant varieties actually express more ergosterol than normal. And this particular antimony worked by generating reactive oxygen species. And so we have published quite a few papers on this antimony and Leishmania interaction, all of which is not possible to present today. So then I wanted to also mention, which I didn't before, after coming to the drug, I wanted to mention that this parasite is not so young, because it was detected in the Cretaceous Burmese amber about 100 million years ago in the gut of a sandfly, which is now extinct, Paleomya burmitis, and also in another 20 to 30 million years ago in Dominican amber, and that is Paleoleishmania neotropicum in the gut of Lutzomia adicatis, another fly. So it is also found in the fossils about 100 million years ago. So this parasite and the sandfly were evolving together. So then it probably originated in the Mesoleishmania, which actually infected dinosaurs. And there's a theory also that maybe many dinosaurs died because of leishmaniasis. So it appeared around the Mesozoic, and then it kept on evolving. In Egypt, in the prehistoric times in Egypt, the several mummies that recovered from the Egyptian pyramids were found to be positive, found to be having Leishmania DNA. So there is actually a record of having this leishmaniasis in ancient times in the Egyptian history. Now this leishmaniasis is spread all over the world. And today, of course, in the context of coronavirus, this has gone to the background. But There are mainly two kinds of leishmaniasis. One is visceral, which kills people if not treated, and cutaneous, which is very much disfiguring and causes skin lesions. Now, PKDL occurs after the visceral leishmaniasis. It's a post-Kala-Azar dermal leishmaniasis, and mucocutaneous leishmaniasis occurs after the cutaneous leishmaniasis has happened. So, and it affects about 1 billion people living in endemic areas, which are at the risk of infection, and frequently infection happens, and especially in immunocompromised people. And with HIV coming in, causing immunosuppression, this has been a real problem of the two. Now, this parasite has a digenic life cycle where it's in the humans in the form of rounded amastigotes and is taken up by the sandfly, and sandfly gut, it's a free-swimming form. Then it's given to another human and goes from there either to dog or to human. And so it is either a rounded amastigote or a free-swimming promastigote that this is the form that it survives. Now, why did we choose to work with the Leishmania parasite? We were working on apoptosis, a programmed cell death in mammalian cells at one point of time. Then it sort of in the macrophages, and also as we thought that is it possible that unicellular organisms have a programmed cell death? And it was in the 2000s, and so not much information, probably no information was available of what happened to these unicellular organisms. So we started working on Leishmania. The reason we chose Leishmania was because it had a single mitochondria. It doesn't have too many mitochondria. And because mitochondria is very important for programmed cell death, we chose this organism to look at it. Now, this is a scanning electron micrograph which shows the form of the form with the tail and free swimming form, and this is the amastigote form that is recovered from inside the macrophages. And this is how it looks under in the in vitro conditions where it is moving there, but in the body they would be moving similarly, I would guess. And where no recordings have been found on what's happening. Although with 2-photon microscope, we can still trace the Leishmania a bit. Now, so mitochondria, as you know, sets of mitochondria control death by apoptosis in cells. There are, of course, mitochondria-independent pathways. But in the Leishmania, as you see, it has a single mitochondrion. And if this mitochondrion actually switched off at once, what would happen? Leishmania will not have the option of death by apoptosis or programmed cell death. So, but they do die by apoptosis. These are the markers, and that are showing that they can undergo apoptotic death if you put oxidative stress. Now, what will happen in the mitochondria? So we looked closely at the mitochondria, and what we saw is that the mitochondria this red means that it is a control mitochondria, which is giving normal mitochondrial potential. But this is a mitochondria which is treated with oxidative stress, and you can see heterogeneous regions of green and red. So green is where the potential is lost, and red is where the potential is intact. And these are the overlapping regions where both are happening. So which means that the mitochondria actually behaves like a group of mitochondria in mammalian cells. And they switch off gradually, not just at once, they do not switch off, they switch off gradually, giving the cell the opportunity to undergo apoptosis is an energy requiring process, and the mitochondria needs to produce a minimal quantity of ATP to sustain the cell till it is actually going to die. So this was one of our very early findings, and this paper was very, very well received at that time. And then later on, we have also shown that not only apoptosis, autophagies were very important for this parasite. And we have generated knockouts of the ATG8 protein, which actually helps the autophagosomes to form. So we did produce knockouts and we tried that autophagy, how did autophagy increase? So autophagy increased in response to oxidative stress. And if you blocked autophagy, parasites were not viable. And then when we generated these knockouts, we actually interfered with the differentiation to infective forms. So ATG8 knockout actually reduced also capability to infect, because if it does inhibit differentiation to infective form, it would reduce the capability to infect, and that's what we found. So both apoptosis and autophagy are very important for this parasite to survive. Now, coming to the host of the parasite, which is the neutrophil, the macrophage, and the dendritic cells. The host, the intermediate host is the neutrophil, and macrophage is the host where the parasite survives within the phagolysosomes. It is very unusual for any pathogen to survive in the phagolysosome, because it is the very compartment which is supposed to kill it. So there are very few pathogens that actually survives there, but Leishmania does. So in the neutrophils, what happens when the fly bites the skin? At the site of the bite, all the neutrophils are called immediately by factors released by the parasite. So once the neutrophils come in, they can set up the traps and kill the parasites, or they can engulf it and kill it by generating the oxidative stress, which is superoxide, the NADPH oxidase, which is on the phagosome, and then that gives hydrogen peroxide and peroxyl radicals and singlet oxygen, and of course peroxynitrites. So the NADPH oxidase actually generates this and tries to kill the parasite or any pathogen for that matter. Now, if in case of Leishmania, if you go to neutrophils, the neutrophils engulf the parasites, and two things can happen. One is that the neutrophil will undergo death and release the parasite, which is more competent to infect a macrophage, or the neutrophils with the parasites can actually undergo apoptosis and be picked up by macrophages. And in this way, the parasite actually hoodwinks the macrophage in taking up the apoptotic cell, but going inside, it ruptures and Leishmania is released. So this is called the Trojan horse mechanism. And this is so it can act as a reservoir, or it can act as a killer cell. So neutrophils have very different roles in macrophages, in leishmaniasis. But I couldn't resist myself to say this, that in case of the coronavirus, what we are seeing, neutrophils are involved in early antiviral, defense can be very, very dangerous, because through their degranulation and lysis, they can be cytotoxic during the severe pneumonia. Or even there is an increased peripheral neutrophil to lymphocyte ratio that actually leads to lung damage in some patients and might involve dysregulated neutrophil activity. Neutrophil traps, which are normally meant for killing the pathogens, go inside the lungs and cause havoc by sending out these traps. So for neutrophils are very dangerous for the coronavirus disease, but we cannot do without neutrophils as well. Now, how do infected neutrophils regulate their own uptake by the macrophages? This was one of our questions. So autophagy, as you know, that the pathogen is taken up and the autophagosomal membrane is formed where it is digested. These pathogens are ultimately fusion with the lysosome leads to degradation, or otherwise, as I said, there is this Trojan horse mechanism. So in our studies, what we showed that when we infect the neutrophils, they undergo autophagy. This red is an autophagy marker, and you can see on the western blots also that the LC3-2 is formed. And this is a control where in starvation you're seeing the autophagosomes also. Now, this is a infected neutrophil which is picked up by the macrophage. So these infected neutrophils actually release the parasites inside. But if you inhibit this autophagy, this uptake by the macrophage is reduced. Here you can see that when we use 3-MA, which is an inhibitor of autophagy, the infection as compared to only infection is reduced. And if you give rapamycin, which you know is an autophagy inducer, actually there is a high uptake of the neutrophils. So the autophagy here is important. May not be the leishmania itself, but leishmania has used this pathway to its advantage. So this was a model that we built, and we actually showed, I mean, I'm not going to go into the details of this, that the uptake of the neutrophil is due to, not due to phosphatidylserine exposure, which is eat me signal, but due to redistribution of do not eat me signal, which is the CD47. So CD47 actually redistributes itself on the surface in response to the autophagy created by the parasite and is picked up by the macrophages. So it is the do not eat me signal that is actually inducing this uptake. So now we come to the macrophage, and it is one of the specialized functions of macrophages is to kill the microbe. Now, why do so many pathogenic bacteria replicate in the very cells equipped to destroy them? But most of them survive in the cytoplasm. But leishmania survives in the phagolysosome. So the competing demands faced by the macrophages actually imply the existence of an evolution, some kind of an evolutionary trade-off, because there are two demands to the macrophage. One is to save itself, and another is to kill the pathogen. So kill the pathogen to save itself, and another to provide housing to the pathogen itself. So it's a very, there is some kind of trade-off in this, of which we understand somewhat, but there are a lot needs to be done. Now, what we did show that it is indeed when the leishmania goes into these phagolysosomes, the red is lysotracher red, and the green is this dye for the leishmania. And here you see, the green is the dye for the reactive oxygen species. And here you see that within the phagolysosome, reactive oxygen species is being produced. Now, if you give oxidative stress to the parasites, the little small red dots are the parasite nuclei, and this is the macrophage nuclei that you're seeing, the complete red. And if you give drugs, you see the parasites are dying within the phagolysosomes by the process of apoptosis, because this green is a signature of apoptotic cells. And how it happens through a calcium-mediated mechanism. This is a calcium signaling marker, and you can see that it is also inside the infected macrophages. So we have published quite a bit on these issues, and but I wanted to just say that at that point of time, we actually showed that wherever leishmania is sitting within the phagolysosome, ROS is being generated, and that is killing the parasite by apoptosis. Now, the three things can happen once pathogen invades. That induction of pathogen apoptosis, meaning that cell is killing the pathogens by using oxidative stress or other mechanisms, or the host cell may want to die because it is unable to kill the pathogens. That is a situation. And the third kind of situation is subversion of the host cell apoptosis. So the pathogens are so smart that sometimes they win, sometimes the host wins. So if they win, they derail the apoptotic pathways of the host. So now, how the parasite manipulates host apoptosis using host signaling pathways? So this is the subversion of host cell apoptosis. In a model, what we did is that we showed that the parasite is very smart. It binds to the TLR2 receptor and activates PI3 kinase and phosphorylates PI3 kinase and phosphorylates ERK. And there are in between procedures that happen here. And then they also activate STAT3. And STAT3 then initiates the transcription of BCL2. Now BCL2 and then the protein BCL2, which is formed, actually does go to the cell and inhibits nitric oxide production through INOS inhibition. Now BCL2, as you know, is an anti-apoptotic protein, and overexpression of BCL2 would give advantage to the host cell to survive, but it gives actually advantage to the parasite producing the BCL2 more and trying that the host cell survives to keep it. And in another, then what it does is also it through PI3K, it activates phosphorylates AKT, and the BAD is phosphorylated and destroyed. So BAD is a pro-apoptotic protein, which is also destroyed. So the cell has lost a machinery that it uses to kill the cells. So a pro-apoptotic protein is gone, and anti-apoptotic protein is increased, which has dual function. It has a cell, it counteracts bad BID on the mitochondrial surface, and it also inhibits nitric oxide. So these are very detailed studies, and but this is one mechanism that the parasite uses to survive within the host cell. Now, how the parasite manipulates host apoptosis using its own defensive enzymes, so using its own proteins. So I'm sure there are more proteins involved in the earlier model I showed you, but that we have not really looked at. So it has two very critical defensive enzymes. One is this is cytosolic tripyridoxine peroxidase, and this is mitochondrial tripyridoxine peroxidase with a mitochondrial signal. And these were built with the information available in PDB, and they exist as decamers within the cells. And this is also modeled on trypanosoma, CTX and PX. Actually, leishmania was not available at that time. And so this enzyme is a terminal peroxidase that generates the radicals and water from this cycling trypanothione, which is like glutathione, but trypanothione is used by this parasite instead of glutathione, and then it is reduced, and eventually the peroxyl radicals are eliminated by this process, and any of the peroxides that are formed. So this enzyme, this cytosolic tripyridoxine peroxidase, we saw that it is actually secreted out. So we wondered that why it is actually secreted out. So what we did was we actually saw that what is the secreted protein binding to in the host. And we found that the secreted protein is binding to apoptosis inducing factor, which is normally released by mammalian cells in response to stress from the mitochondria, and then it leads to DNA degradation after it moves to the nucleus. So then we did a in silico study, and we saw that actually the tripyridoxine peroxidase binds to the apoptosis inducing factor through their fat binding domain. And so there are many residues that can be identified through it binds. But then we confirmed it by immunoprecipitation studies using both tripyridoxine antibodies and the AIF antibodies, and it was shown that they actually bind to each other. Now, what is happening here? What is happening here is that the parasite, with the tripyridoxine peroxidase being secreted by it, it actually goes into the cell. There also inside it secretes the tripyridoxine peroxidase, and while the host cell tries to die by sending the AIF to the nucleus from the mitochondria, they bind to the AIF in the cytosol. And once they bind, they're prevented from going to the nucleus and killing the cell. So this is a very efficient way by the parasite by which it actually comes inside and secretes this protein, or even in the surrounding media. And we have shown that it can be picked up from the surrounding media. And so this is a way by which the parasite also survives, by preventing the cellular apoptosis to happen. So if you just look at this cell, this photograph, this microphotograph shows that this is a nucleus and these are the AIF in the cytoplasm. Once you give staurosporine actually sends them to the nucleus, means it's a killing signal, which makes the AIF to go to the nucleus. And so it happens if you have a control cDNA. But if you have overexpressed CTX and PX inside the cellular cytoplasm, then what happens is even in the presence of staurosporine, this AIF do not travel to the nucleus to kill the cell. It's a very visual. This is a very recent study that we have done. And it's very exciting that this can happen like this. So now, so if I try to, this data, if I try to summarize, it'll be like that the parasites, you can kill them by inhibiting the, with antioxidants, you can actually sort of make them sick. And with oxidants, and if antioxidants we used to see how the parasites would react and they would thrive. So oxidative stress is a very important thing with the leishmania. It has been shown with many pathogens. And then we have used BH3 mimetics, which means they are BCL2 inhibitors. So the model I showed you, there we have used BH3 mimetics, which are available and are used for cancer therapy. And these BH3 mimetics can be, we have tried it in animals also, and it works. They can be repurposed for the purpose for the treatment of leishmaniasis, but they're very expensive. And now in the context of the current situation, these are all have taken a backstage. Now, anti-autophagic agents, which are also in trials in cancer therapy, they can be used to stop autophagy in the neutrophils and prevent their uptake by the macrophages and reduce the parasite load. And of course, the TXN, the dipyridoxine peroxidase inhibitors, can be used to reduce infection. So we have actually, our studies have pointed out multiple points at which inhibition can be made and drugs can be repurposed. But unfortunately, now we cannot do this as scenario has changed, and research preferences are going the other way. Although academic interest will always be there, and we should always be aware that even if leishmania is eliminated, there is always a danger for it to come back. So I wanted to make a few comments in last two slides that this before the COVID-19 struck, the global travel and deforestation and the traffic of the international blood products, global warming, etc., were affecting the leishmaniasis distribution, and it was occurring in places where it is not supposed to occur. So, like for example, there was a huge displacement of refugees from Syria to Jordan, and that brought leishmaniasis here. And this is primarily cutaneous leishmaniasis. Visceral leishmaniasis are also there, but the cases of cutaneous leishmaniasis are more. And it was largely confined to areas around Aleppo, but they came to Syria. They came from Syria, and because it was a war in Syria, so the conflicts are creating the distribution, the change in distribution of these diseases. And it had a typical relationship with the civil war in Sudan, where immediately following a civil war there would be a surge in infections. So because there would be compromised health programs, medical care shortage, destruction of health-related infrastructure, and all kinds of things would happen in a disturbed situation. And therefore, and even today, I think these unfortunate people who are in these areas who are suffering from these crises that we see are really affected. So in the elimination program for the leishmania, they were very good. I mean, Nepal has done it very well. Bangladesh has done it very well. And in Bihar, about more than 70% of the endemic blocks have achieved the elimination target, which is very good. But it is due to the vector control, the sandfly control, that is very effectively done. Then rapid diagnosis of the disease and the treatment given. And this time, mostly miltefosine is given, which is also a repurposed cancer drug, and the follow-ups and due to very, very dedicated health workers. So it has reduced in most of the parts. While it remains as a parasite, is a very interesting point of study, and it can emerge anytime coming back. This is one of the success stories of elimination. Like if you read the story of smallpox elimination, it's a very beautiful story how smallpox was eradicated from the world. So like that, leishmaniasis is also being almost eliminated. It's confined. This is visceral leishmaniasis I'm talking about. Cutaneous leishmaniasis is still found in Africa and in South America. South America, it is quite prevalent, so they need to work on it more. So these were my soldiers, who some of them, I think most of them, who worked for me, and together had a very nice laboratory conditions we worked, and all jolly good fellows. And then, of course, the grants that came to support our activities. And I could give you a flavor of kind of thing we do. I couldn't elaborate on many things, but I hope I have given you some idea of how host pathogens can interact and create difficulties for us. Thank you very much. I'm not sure what time I took, but...
No, ma'am, it's perfectly fine. Thank you so much, ma'am, for this wonderful lecture. We have a few questions, so if you allow, we can take some of the questions.
Sure.
Okay.
So I'll stop sharing.
Yes, ma'am, you can stop presenting.
Yeah.
Okay, so the first question is from Rohan Pawar. He wants to know that what is the stand and accuracy of computational biology studies in the domain of pathology?
This computational biology has come in a very big way, and accuracy is, of course, depends on what programs are you using and who is using that, and this big data thing is going to come in pathology in a big way, because that will help us to generate algorithms by which we can predict many things. But like in everything, there is some kind of, it needs a lot of, what I should say is rigor. It requires a lot of rigor. But it is very much there, and it is going to happen.
Okay. The next question is from Shanchari Godai. How could they detect leishmania in the mummies of ancient times?
Yes. See, from the ancient remains, it's a very it's a science now to detect this DNA for leishmania. Not only leishmania, for many things we can now find out by our little PCRs that how this organism may have been present over there. So it's a science by itself now. So you extract the DNA out of DNA, it stays in many things. So you can extract the DNA from the tooth that were preserved in the mummies, and that DNA you can then amplify, and you can then match it in the database with what it was. Not only it's leishmania, it can be anything. And so it's the pulp of the teeth or something that remains there, or in some part of the bone where it is preserved. Even I'm talking about only the mummies, but in the fossils, the fossils I showed was done like this because it was DNA was preserved there.
Okay, the third question is from Vaisak Vishwanath. Do antifungal drugs have any broad range effect on protozoan parasites?
Yes, they do. They do. See, one of the main drugs for leishmania is amphotericin B, which is an antifungal drug. So, but it's the only thing is it has to be given in a big dose, and it creates a lot of side effects. So, antifungal drugs have been tried. Ketoconazole, and there are many of those antifungal drugs that has been tried in leishmania. But because of the requirement of such a high dose, it has never been really brought into practice, except this, which is ambisome, which is now a formulation of amphotericin B, which is supplied inside liposomes and given. And that gives a better, that causes lesser side effects. So yes, antifungal drugs are suitable.
Shubhi Dasgupta wants to know, from an archaeological standpoint, does leishmania play an important role?
Archaeological standpoint, I'm not exactly getting the question. Archaeological standpoint meaning It's found in mummies. It's found in in the fossils, and it helps to I guess this kind of studies helps to trace where they came from, when they came from, what was the pattern of evolution. So archaeology in its sense, it also determines what were the vectors or what were the parasites at a given time point in our history. So that's the way it may be important for the archaeological and archaeological research. You find these civilizations, like our Indus Valley civilization, or the different civilizations that you can find materials where you can dig out a DNA and find that okay, at that point of time the likely vectors that were present are these. You will detect some, you will not detect some, because always you will not find the DNA everywhere. But it's important in that context of knowing the history.
So Pradeep Kumar Vishwakarma wants to know how mitochondria controls death by apoptosis. And another question it's kind of related is mitochondria directly affected by oxidative stress or any other mechanism present behind it?
Mitochondria itself generates oxidative stress because it has the respiratory chain, so it generates many radicals itself. So it can produce oxidative stress within the cell, but like any other cellular organism, it can also be affected by oxidative stress, which can come from outside. So mitochondria itself is susceptible to oxidative stress, but the way it creates cell death is that mitochondria has cytochrome C, which is a part of the respiratory chain, and that gives us life. This cytochrome C actually gives us life, but if the mitochondrial membrane is breached, the cytochrome C comes out and it becomes lethal. It combines with different apoptotic factors, and it activates caspases and creates cell death. So it forms apoptosome, and then it creates the processes that lead to death through chewing up of the cellular substrates. So mitochondria, the main thing is that cytochrome C, whether it is in or out, actually makes a difference between death and life of a cell. So this cytochrome C release happens by opening up of mitochondrial channels or breaking of the membrane, because this cytochrome C is limited within the mitochondria because it is required for the respiratory chain.
Bishwadeep Ghosh Roy wants to know, is there any caspase activation or T cell mediated response when leishmania invades the host cell?
Yes, there is. Caspase activation. See, there are two death pathways. As I said, one is this mitochondrial death pathway, other is the non-mitochondrial death pathway. So we have found caspase activation in other cells in different conditions. That caspase activation do happen, and at that time the AIF translocation may happen or may not happen. So it depends on which situation. It's always like this in the apoptotic system, that it may be a caspase independent death, it may be a caspase dependent death, it may be a death which is combined. So it can happen that way. What was the second part of the question?
This caspase activation and T cell mediated response.
T cell mediated response is there once it invades the body. The antigen presentation takes place from the dendritic cells, from the site of the wound, and then it goes into the body. And the dendritic cells would take it to the different lymph nodes and expose the T cells to this leishmania antigens which are expressed on its surface. So the T cells, if they're memory cells, they will remember that whether the body has been exposed to this pathogen or not, and so they will proliferate those memory cells. Otherwise, the T cell will mount a response against this parasite. It's like any other pathogen. It happens, it mounts a response and tries to kill the pathogen by various means, like killer T cells will go and try to kill the pathogens, macrophages will try to engulf them. So a host of different things will take place once the T cell gets sensitized by the dendritic cells.
Shondi Pramanik wants to know why does macrophages show reduced number of parasites under a BCL2 deficient background?
Why does macrophages show less
number of parasites?
So what happens is once the parasites go in, they try to increase the BCL2. Why do they try to increase the BCL2? Because the BCL2 is anti-apoptotic and prevents the cell from undergoing apoptosis. So what happens is when they go in and the cell is trying to die, if there is a deficient BCL2 deficient background, then the cell is trying to die, and when they're trying to die, under the altered metabolic conditions, the parasites cannot survive there. So it is not that it becomes all or none. There are few parasites who can survive and most of them will not, because of the altered metabolic condition that is happening due to the BCL2 deficient condition.
Okay, and there's Shopto Banerjee wants to know, can you tell us the status of AIF in different breast cancers?
No, I will not be able to tell because I'm not updated on that literature.
Sitaram Maharana wants to know, is GP63 protein of leishmania, does GP63 protein of leishmania induce apoptosis?
GP63 and LPG has very different roles with the host cell. The LPG often tries to reduce cell death, actually, the LPG. So we have used LPG knockouts to show that if there is less LPG, then the parasite is less successful in doing infection. Now, if for the GP63 also it's the same case, because GP63 helps the parasite to make its niche within the macrophages. So if there is less GP63, there would be less survival of the parasite within the cells. So there are some studies where they have done some kind of inhibition studies with GP63.
Dr. Priyanka Dev wants to know, we have heard that leishmania genome plasticity allows swift adaptation to new antiparasitic drugs. So what is your experienced viewpoint on targeting leishmania host cell interactions to circumvent leishmania drug resistance?
Well, yes, leishmania, became resistant to this antimony very early, when even in the early years, and when they started mixing it with other heavy metals like arsenic and all. So it is really efficient in generating a drug-resistant form. And if you use multiple drugs together, a cocktail of drugs, or even two drugs together, you reduce the probability of leishmania to get resistant. And primarily why it gets resistant is because especially with antimony, is that it got converted to the trivalent antimony inside the parasite, and it got thrown off by the pumps, by the drug elimination pumps, and that is why it got resistant. So there are multiple mechanisms that it uses to have the drug resistance.
Can we take a few more questions?
Is there time? I can.
We have no problem.
Yeah, sure.
Okay, so Shrikant Borubala wants to know, please explain about visceral leishmaniasis and is it fatal?
Visceral leishmaniasis, if it is untreated, it is fatal. It'll eventually, what happens is the people die because of anemia, because it affects the liver, it affects the spleen. So because of the engagement of these two organs with leishmaniasis, there is severe anemia, and people mostly die because of that anemia created by the bug. So visceral leishmaniasis, if you don't treat it, people will definitely die. And this is why I told that the villages after villages got, eliminated. So many people died. And it was only when this antimony came in, Upan Brahmachari actually saved thousands and thousands of lives by creating that antimony concoction that he made.
So another question from Sitaraman Maharana is: are arabino substitution in LPG and serum concentration help metacycling promastigote survival in the high acidic medium of phagolysosome?
This is a very complicated question because I do not know because I have not worked with arabinos and I don't know if people have really tried to do it, but so I won't be able to answer this.
Mm-hmm. And Dr. Anupam Ghosh wants to know, would you please shed some light on the prevalence of leishmaniasis in India?
In India, as I said, that the leishmaniasis, it's very interesting that leishmaniasis is mainly confined to Bihar in an endemic zone in Bihar. There are some foci in the West Bengal and Bangladesh, that is of course outside India, that these leishmaniasis occurs. But recently we have seen cases in Rajasthan of these, but these are caused by the same parasite, but they're not the visceral leishmaniasis. They're post-kala azar dermal leishmaniasis. So you get it on your hands and mouth and as skin lesions. So it is a parasite that caused visceral leishmaniasis, but in the area of Rajasthan we get mostly PKDL. So why PKDL is caused by the same parasite? We do not know if climate is related to that. We have no idea, but it does cause a disease like that. So it is very important that we know why the same parasite. See, there is a cutaneous form of leishmaniasis where the parasite, other species of parasite causes it. But here we have the same Leishmania donovanae, which causes a visceral leishmaniasis, would go and cause PKDL in some other part. And PKDL will come once the people actually suffer from the visceral part. So this is what now people are looking at, is what is happening with the visceral leishmaniasis and why it is coming on the skin. So it happens that in India we have in Rajasthan, we have in Bihar, and we have in West Bengal, but there is no other place in India that we have leishmaniasis.
Leo Rodriguez wants to know, where can I test plant extracts for anti-leishmaniasis?
You have to have a laboratory who works on leishmaniasis, and there they can test the plant extracts in vitro cultures with metacyclic promastigotes, or you can get amastigotes out of the macrophages. It can be tested. So that laboratory has to be competent to generate amastigotes, and because amastigotes are the disease-causing forms. So the most effective use of anti-leishmanial would be on the amastigote stage. And the promastigotes also, they come when the sandfly bites the human. So, or when they leave the body to be picked up by the flies, but mostly it is the amastigotes that need to be targeted. So, and they are physiologically quite different, the amastigotes and promastigotes. So you have to find a laboratory where they will be willing to do this, and also have a animal model, because people have to have an animal model to study any drug for that matter.
So we'll take one last question. Babu Moni Ram wants to know, does the parasite induce autophagy in the cells or hijack the inherent autophagosomes? And are there any receptors involved?
Interesting questions. So what we see is that when we put the parasite, there is an autophagy increase. Now that happens in case of neutrophils, that happens with many pathogens. It's not only that the leishmania will create an autophagy. So I would say that it is the inherent process, housekeeping process of autophagy that is being influenced by the parasite itself. So it is the inherent autophagic process. The parasite cannot create autophagic process inside by, unless it has the cells help to create that.
Okay. So I think we should stop now, because There's not enough time. I'm sorry if all the questions were not answered, but we have limited time. So, ma'am, thank you again for taking the time to be with us today and for this wonderful talk. I would now like to request our convener, Professor Renare Banerjee, to give the vote of thanks and close the session. Ma'am, over to you.
Thank you, Shinjini, and thank you, Chandrima. That was a wonderful talk. I enjoyed it so much, and as I'm sure our other viewers also would have, because I see a lot of questions, and still a lot of questions are there. We would be happy to pass it on to you. But now,
send it to me.
So
Send the question.
Sure. I will do that. So thank you very much for a very interesting talk, because as you rightly said at the beginning of your talk, that these microbes were there before we came and will remain after we are gone. So it is a question of human resilience, especially by the thinking power of the humans to make up for what their body probably, our body probably is not capable to do. So there will be that story of armament and counter armament with our adversaries, planet Earth. So the story will continue, as they say. So in conclusion, I wanted to talk a little bit about, for the viewers, those who don't know, Professor Shaha actually played cricket 1975 to 1977, right, Chandrimadi? So
73 to 77, actually.
73 to 77. So, as a Gokelite and a Brabornite and a woman scientist, a fellow woman scientist, may I ask you what your message to the budding women and aspiring scientists would be? Because your talk would be archived in our YouTube channel, and many more viewers than the few hundreds who are listening into you now would be tuning in and listening to your answer. So we both know that still a lot of gender disparity exists, especially in higher education, and it is tough. So what from your journey would you like to share with us as women, as scientists, as strugglers in a world that is still revealing itself?
Well, I have, in my journey, I have come across many women students, and they are the ones that eventually leads to the leadership positions or to some, some other positions. But I have found that there are, depending on how they are brought up, they react to the situations. And so I ask parents that bring up your daughters like your sons, because if you are given, if the daughters are given that you cannot do this, you cannot do that, and you should not be doing this is not your subject, biology is the only thing you can read, and it's the parents' duty to sort of give them independence and tell them that you can do whatever you want to do. And so there comes, biologically also in a life, there comes a time when people, they get married and they have kids, and at that point of time they need a lot of support. And that support is not only from the family of course has to support them very strongly, that they shouldn't lose out on that time on in their careers. And the working place also has to give them support, because we do not have very women friendly features, I should say, in different institutes or universities, that you don't have crash, you don't have a separate room, you don't. So those are the things that are needed very much. So joint support by the family and the workplace is very essential. And I personally think that there is a lag which we have to recover, because many of the students have lost out somewhere. There is this leaky pipeline, we call it, and they have lost out somewhere. They have just left doing science, and went into doing families, and then did some other odd job. So they should also by themselves should have a very strong focus on what they want to do. Now, as far as science goes, I think they have to really love doing it and not take it as a job by itself. It is only then that they will be able to continue with the support of their families, with the support of the laboratories that they work in. And, more and more things are becoming possible because our Department of Biotechnology, Department of Science and Technology have many women-friendly programs. I don't really have an idea how successful they have been because there has been no evaluation of that. But I think when you are growing up, if your parents give you the opportunity to explore yourself, that, okay, I want to do this, and that I'm not restricted if I want to do something. Suppose somebody wants to go to North Pole, the parents should not say that you can't go. So, being a woman, it becomes like that. But I think, things are improving. And this disparity to self-correct will take a very long time. So we are trying to take active actions so that we can really help people to cross that barrier and come forward. So I think it's happening, but it's taking. But the mindset has to change in many women. There are two classes of women. Some are very good, think that I have to go ahead. There are some who gets inhibited by the way they were brought up, that they cannot help. So I think parents are to be taught that you should bring up your daughters like your sons. So.
Indeed. So thank you so much. Thank you so much, and my best wishes to you. And I hope we meet.
Sure. Let this be over. And
let this be over. Yes.
Well, hopefully.
Okay.
Yes. Thank you.
So thank you, everyone, for tuning in. And with that, we are signing off for tonight. Bye-bye.
Thank you. Bye.
Thank you, ma'am. So join us tomorrow again for our regular lecture by Dr. Prabhavan Chakraborty. And please follow us on Facebook and Twitter and subscribe to our YouTube channel. Our previous talks are all archived in our YouTube channel, so you can check them out there. And thank you again, and take care, and see you tomorrow.